Are there any questions? All right, I'm not hearing anything. So let me state, I've got some grading done. I haven't quite finished the last lab. Most of you are graded, but there's like 4 students I need to get them graded. And I got some other assignments graded and I don't remember what it was. So take a look at the your grade book. I've got some things graded. And with the lab, remember that on the first grading of the lab, you will have about 5 to 7 days to make a correction if you didn't get 100%, and you can submit the corrections for full credit. So you do have 2 attempts in the lab, and I will grade it the first time so you know what's wrong and you all you need to do is make corrections. Any questions about any of that?
Yeah, quick question. That one quiz that you had to kind of cancel, is that still affecting our grades, and will we not see it adjusted till the end?
I'm not sure. I haven't canceled any quizzes. I did
It's
remove
it's
quiz
the
like 2B or something like that because you're
yeah.
supposed to take 2C instead. Everybody took 2C, so your grade will be on 2C. I've changed the value of quiz 2B to be 0. I can't delete it because somebody got in there and and they took the quiz and they had a score. And so I made it out of 0. And that student, I think, let me check on that. I think what I did with that student is I made them a 0 on that quiz. Let me check to make
Okay.
it's right. Oops,
I
that's not.
just wanted to make sure the grade I see is that my actual grade.
It should be. Okay.
Okay, thank
Well,
you.
let me make sure. I think I'm sharing this. Let me stop that. I can't get into the grade and share that. Let me make sure. Oh, shoot. I do remember I made quiz 2B worth 0 so that it won't add to your grade or subtract from your grade. But then there was one student who got a score in it. And I'm not sure that what I did with hers. So let me take a look. Hmm. I must have made that quiz oh, what do you call that? Because that quiz isn't in the grade book anymore. So I must have made it a practice quiz. Whoops, that's not what I wanted. Let me go into the quizzes. Oh, there it is. Well, it's not in the grade book, so you're not getting any points for that. But Lindsay did, I guess I shouldn't have said that, but let me check in here.
Lindsay speaking. My grade, I was the one that took the quiz. My grade is accurate right now.
Okay. Like I said, it's a practice quiz, so I don't think it's in the grade book, so it's not going to show. Let me just. Well, I'm not going to be able to switch this to 0. So let's just say, hopefully everything's correct. And I'll make a note to check on that because it's taking too long.
I'll just email you separately with my questions.
I've got a note, so that's fine.
Okay.
If anyone else has any other questions, please go ahead and ask. I'm actually was in the grade book. I don't need that. So I close that. Let me come back to my screen. All right, let's share my screen. Let's say today is October 24th, and we're supposed to be working on chapter 4 and finishing chapter 4 today. And then in the lab, we'll be discussing the unknown project, and hopefully we'll get that started today too. Any questions about what we're doing? If not, let's move to chapter 4. The last slide I talked about on chapter 4 is shown now, where we were talking about the plasma membrane. And we mentioned that the cell membrane is actually a liquid lipid layer. It's a bilayer, mainly composed of phospholipid, and it is as fluid as olive oil. The cell membrane, oops, let me go back and delete that too here. The plasma membrane shows selective permeability. This means it allows the passage of some molecules, but not other molecules. No large molecules can move across the plasma membrane unless they have a transport protein that can move the molecule across the cell membrane, but they cannot diffuse across the cell membrane. Ions either do not move across the plasma membrane, or if they do, they move very, very slowly. Lipid molecules can move directly across the cell membrane, but if you think about it, like a blood cell in the blood surrounded by mostly water, you're not going to be coming across a lot of lipids in the blood. There are some, I'm not saying there's none, but most of the molecules are going to be water soluble because the blood is water soluble, or has mostly water. And most lipids are not soluble in water. So there can be lipid molecules moving into the cell, but generally there are not that many lipids that would, because outside the cell is mostly water. And then small molecules can diffuse across the cell membrane. Mainly that's oxygen and carbon dioxide. Water can move slowly across the cell membrane, and I'll try and show you that. I don't think I've discussed that, have I? What happens is, let me blow this up with water. This phospholipid can move. Let's say it moves over there. Water can then move in, occupying the space, and water is much smaller than a phospholipid, but you're getting the point. However, the water cannot move through the next lipid bilayer, and the water just stays there. And that keeps a hole in the phospholipid membrane where the water is. And the water just moves, waits until this phospholipid moves elsewhere. And then the water can move across. And then when it moves across, usually the phospholipid moves in and then closes that hole that was on that side of the membrane. So water can move across the cell membrane, but not very rapidly. It's much too slow for, let's say, green plants and the roots to absorb water. So many cells, especially plant root cells, have aquaporins, which are transport proteins that actually move the water across the cell membrane. And aquaporins are in actually many different cells, not just plant root cells, but I know that aquaporins are in plant root cells for that. But the small molecules of oxygen and carbon dioxide can simply diffuse across the cell membrane. And that's important to the cell because it does need oxygen, and it just diffuses across the cell membrane into the cell. And carbon dioxide, which is produced by the cell, just diffuses across the cell membrane. And then it's taken up by the blood. The blood will carry that carbon dioxide to the lungs, and then the lungs will breathe it out. And of course, the oxygen is breathed in by the lungs and then carried by the blood to the cell. Any question about any of that? All right. In the plasma membrane, we do have a few other things going on, especially in prokaryotic cells. In prokaryotic cells, this is where we find the enzymes for making the majority of the cell's ATP. And we'll talk a little bit more about that in a future lesson. For the photosynthetic bacteria, this is also where we find the photosynthetic pigments. They're in the plasma membrane. And what I really should say, they're in foldings of the plasma membrane. Let me blow this up as much as I can. So in this photosynthetic bacteria, the plasma membrane is not running parallel to the cell wall, which is here. The plasma membrane runs this way, and then it folds, and then comes back, and then moves here, and then folds. And in these foldings, we have the photosynthetic pigments. These foldings are called chromatophores or thylakoids, probably after what was that called in the chloroplast, the thylakoids? No, that's not right. Anyone remember what that folding is in the chloroplast? Let me end this. Yeah, the thylakoids. So that's where the photosynthetic pigments are in the cell membrane, or foldings of the cell membrane. And the same with the enzymes that make ATP. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and then the polymyxin antibiotic. Each of these, if just a little bit of alcohol, will put a hole in the membrane. The polymyxin B will put a hole in the membrane, and a little bit of quaternary ammonium compounds will put a hole in the membrane. That will cause leakage of the cell contents, the cytoplasm, to leak out, which will kill the cell. Of course, if you use a lot of alcohol on the cell, you'll just dissolve the entire cell membrane. So it'll disappear. All right, let's talk about the movement of materials across the membranes. For bacteria, it's only across the cell membrane, but for eukaryote cells, it's across any membrane, like the nuclear membrane or the membrane of the chloroplast or the membrane of the mitochondria. There are two processes used to move materials across membranes. There's a passive process and an active process. The passive process is where a molecule moves down its concentration gradient. So if the concentration of the molecule is higher outside the cell, it can move into the cell down its concentration gradient. This costs the cell no energy because the molecule is moving in its normal way down the concentration gradient. There are three possible ways to engage in passive movement. You can have simple diffusion, where the molecule just moves from areas of high concentration to areas of low concentration. And that happens with oxygen and carbon dioxide. They move across the cell membrane simply by simple diffusion. There's also facilitated diffusion, and that's where the molecule is moving from areas of high concentration to areas of low concentration, but it's requiring a protein channel to allow that molecule to move across the plasma membrane. And that's why we call it facilitated diffusion. The movement is facilitated by a protein. Both simple diffusion and facilitated diffusion is the movement of a solute molecule across the membrane. A solute is a molecule that is dissolved in a solution or in a solvent. So solvent in most cases would be water. The solvent is just a liquid that dissolves other molecules. And then the other molecules are called the solute. Osmosis, on the other hand, is the movement of water across the plasma membrane. Osmosis is the movement of a solvent across the plasma membrane. However, the solvent moves the same as the solute, meaning the solvent moves from areas of high concentration to areas of low concentration by diffusion, although osmosis is diffusion of water across a membrane. It can be a fake membrane, like a, what do you call it? Like a plastic membrane, or it can be a cell membrane or something like that. Water is so important, the movement of water into the cell, that we make this special term osmosis for it. Any question about any of that? All right. All three of these processes do not cost the cell any energy because the molecule is moving from areas of high concentration to areas of low concentration. However, if the cell moves a molecule from areas of low concentration to areas of high concentration, that is an active process. This costs the cell energy because it costs energy to move the molecule from areas of low concentration to areas of high concentration. And that could be something like an amino acid outside the cell is lower in concentration outside the cell than inside the cell. But the cell will see that amino acid and want it. And so it'll pump that amino acid into the cell by an active process. How it will do that is there will be a protein transport in the membrane, which will take that molecule and then pump it into the cell. That will cost energy because the molecule is moving across against its concentration gradient. And the cell will spend one ATP molecule to move one molecule into the cell by an active transport. And the ATP will be broken down into ADP and phosphate that will release the energy that's needed to pump that molecule into the cell. Any question about any of that? All right, let's talk a little bit more about passive processes, the movement of molecules down their concentration gradient. Simple diffusion is where a solid moves from areas of high concentration to areas of low concentration, such as when we put a drop of dye in this beaker of water. The dye will be the solute in the water, which is the solvent, and the dye will move from areas of high concentration to areas of low concentration by simple diffusion. With enough time, that dye will be evenly dispersed throughout the water, and obviously there will be no more simple diffusion, but my point is, given enough time, the water will be all a light purple from the dye diffusing into the water. As I stated, for cells, it's important for simple diffusion to move oxygen into the cell and carbon dioxide to move out of the cell. Any questions about any of that? All right, facilitated diffusion is similar to simple diffusion, except that facilitated diffusion always involves a membrane. Simple diffusion does not require a membrane. It may, like when we're talking about oxygen moving into the cell, there is a membrane there, but this dye in the water does not have a membrane. Facilitated diffusion always has a membrane. The molecules move from areas of high concentration outside the cell to where there are lower concentration inside the cell. And how that happens is there's a protein that moves or allows the molecule to move through the cell membrane. This is called here a transporter protein, and I'm just going to call it a channel protein because it does not actively pump the molecules into the cell. It doesn't cost the cell any energy. And the example of glucose is a bad example because glucose doesn't always act this way. Glucose is really complex on how it acts, and initially it will move into the cell by facilitated diffusion when the concentration of glucose is higher outside the cell than inside the cell. But once the concentration of glucose equals the concentration inside and outside the cell, glucose will no longer move into the cell by facilitated diffusion. The cell will want the glucose into the cell, and then this protein changes from being a channel protein to an active transport protein, and then it will actively pump the glucose into the cell until there's no glucose left outside the cell. And like I said, glucose is a bad example because this protein doesn't always act as facilitated diffusion. Sometimes it acts as active transport. So I want to change this to fructose. Fructose only moves into the cell by facilitated diffusion. So when you eat fruit and you got a lot of fructose in your gut, the fructose will move into your cells by facilitated diffusion. But once the glucose inside your cells equals the glucose outside your cells, no more fructose will move into the cell. Any questions about that? So that means when you consume fructose, not all of it will be digested. And that's different than if you consume glucose, all of it will be digested. Any question about any of that? All right, with both simple and facilitated diffusion, it's the solids that move and they transport down their concentration gradient, meaning the solid molecules move from areas of high concentration to areas of low concentration. When we're talking about water, it is not a solute. It is a solvent, but water also can move down its concentration gradient. And this we call osmosis, the net movement of water across a selectively permeable membrane. And water only moves by osmosis from areas of high water concentration to areas of low water concentration. So when the water is higher, the water concentration outside the cell is higher than inside the cell, water will move into the cell by osmosis. So what happens in the plant when there's more water in the plant than there's water in the soil? Can anyone answer? Well, does water move into the cell? You guys are really not cooperating here. All right. Water would not move into the cell because water moving by osmosis only moves down its concentration gradient. So no water would move into the cell because the water outside the cell is lower than inside the cell. In fact, we will get water leaving the cell by osmosis. Any question about any of that? Now, it's important to realize that when water is moving by osmosis and the water is moving either into or out of the cell, we discuss it as the water moving, but you must realize that the tonicity of the solution is defined by the solute concentration, not by the solvent concentration. So when water is higher outside the cell than inside the cell, that means the solute concentration outside the cell is lower than inside the cell. And we define the tonicity by the concentration of the solute. Any question about any of that? All right, so a bacterial cell can find itself in three kinds of osmotic solutions. There's an isotonic solution, a hypotonic solution, and a hypertonic solution. You should note that each of these are relative terms. So you shouldn't say the cell is in an isotonic solution unless you mean that the solution outside the cell is isotonic to inside the cell. Any question about any of that? All right, isotonic is the easiest one to discuss. In an isotonic solution, remember tonicity is defined by the concentration of solute, even though it may be the water that is moving across the membrane. In an isotonic solution, the concentration of solute outside the cell is equal to the concentration of solute inside the cell. So that means the concentration of water outside the cell is equal to the concentration of water inside the cell. That means there's no net movement of water into or out of the cell. What do we mean by net movement of water? Anyone?
The overall movement?
Yeah, the overall movement. That doesn't mean that you can't have a water molecule moving into the cell. If one moves into the cell, we then have another water molecule inside the cell leaving the cell. And then there's no net movement of water. So if one moves in, then one has to move out to get an isotonic solution. The cells are happy in an isotonic solution because they will not expand by water moving into the cell with the cell membrane expanding. And if you have no cell wall, the cell membrane will continue to expand until the last water molecule moves in, and then the cell membrane expands a little further, and that causes the cell membrane to pop, and that will kill the cell as the cytoplasm then leaks out of the cell. Another thing that can happen is water leaving the cell, in which case the plasma membrane will shrink away from the cell wall. And if the cell is sensitive to desiccation, that will kill the cell. Given enough time, that will kill the cell. So the cells are happy in an isotonic solution. For example, your red blood cells are in an isotonic solution when they're in the blood. You can also find a cell in a hypotonic solution. This is when the concentration of the solute outside the cell is lower than the concentration of the solute inside the cell. This means there's more water outside the cell than inside the cell. So there will be a net movement of water into the cell. That will swell the cell membrane and the cell. If the cell has a cell wall, the cell membrane will expand up until the cell wall. But once the cell membrane reaches the cell wall, it cannot expand any further. That will limit the movement of water into the cell, because once the cell membrane has expanded all the way to the cell wall, there's no room for more water to move into the cell. So water will stop moving into the cell. On the other hand, if the cell does not have a cell wall, the cell membrane will continue to expand until it pops from the water moving into the cell. Any questions about any of that? All right, bacteria often find themselves in a hypotonic solution, like bacteria causing us an infection. And that's actually why the enzyme lysozyme on your skin works against bacteria, and also why the antibiotic of penicillin G will work. Penicillin G will damage the cell wall, allowing the cell membrane to protrude out of the cell wall. And then more water can flow into the cell, because the cell is in a hypotonic solution, until the membrane expands so much out of that hole of the cell wall that the membrane like I said, most bacteria actually do live in a hypotonic solution, and the only reason why they survive is because of the cell wall. Any question about any of that? All right. A cell can also find itself in a hypertonic solution. This is when the concentration of the solute outside the cell is higher than the concentration of the solute inside the cell. This will cause water to leave the cell, because there's more water inside the cell than water outside the cell. This will lead to the cell membrane pulling away from the cell wall and shrinking, which we call plasmolysis, when the cytoplasm and the cell membrane pull away from the cell membrane. This generally leads to death. It does depend on how sensitive the cell is to desiccation before they will die. Like E. coli cannot survive very long in a bottle of molasses because the molasses is a hypertonic solution. And so almost all cells that fall into the molasses will undergo plasmolysis. For most of the gram negatives, that will kill the cell within, I don't know, a few minutes, a few hours, something like that, not very long. However, if the cell has an endospore, or is an endospore, that cell can survive for a very long time in a hypertonic solution. Any question about that? All right. So the movement of materials against the concentration gradient is the next topic we need to talk about. Passive processes are okay when the concentration of the substance required by the cell is higher outside the cell than inside the cell. But what happens when a cell is in an environment with low nutrient concentration? For example, a cell in the open ocean. There are very few nutrients in the ocean water. Or a cell in the Sahara Desert. Very few nutrients in the sand of the desert. So what does the cell do if it runs into a nutrient molecule? Well, it will move the molecule into the cell against its concentration gradient using an active transport. And that way the cell can accumulate nutrients against the concentration gradient. To move a molecule against its concentration gradient, that will cost the cell energy because the cell has to pump the molecule against its concentration gradient. And cells use an active transport protein to pump the solid molecule they want against its concentration gradient. The transport protein will be in the cell membrane, and generally there is one active transport protein for each molecule the cell wants to move across the cell membrane. For example, the cell has an active transport protein for glucose, an active transport protein for lactose, an active transport protein for almost any molecule the cell would want. And then that one active transport can only, generally, can only move one molecule or transport one molecule into or out of the cell. And so the cell makes many different active transport proteins to move all the molecules the cell would want inside the cell. So transport proteins are generally molecule specific. We will talk about a few exceptions. We have some drugs which can mimic the solute that's transported by the transport protein, but generally a transport protein moves one and only one molecule. Like I said, there are some exceptions, but generally that's true. And then to move one molecule into the cell that the cell wants, that costs the cell one ATP molecule. Any questions about any of that? So active transport happens in both prokaryotes and eukaryotes. And I did mention that glucose is an active transport protein, and the cell will continue to pump the glucose into the cell against its concentration gradient until there's no glucose left outside the cell. Why you'll always digest all of the glucose and the sucrose that you consume. All right, let's talk about group translocation. It's a special form of active transport that is only found in prokaryotic cells. Active or group translocation is not found in eukaryotes. It does require a transport protein. The molecule does move against its concentration gradient. So in all of that sense, it's similar to active transport. It is a little different though, and that is it does require a high energy phosphate compound. However, this is not usually ATP. Oftentimes it's PEP, which is just, we're only going to call it a high energy phosphate compound. And then the molecule that is transported in group translocation, the molecule is chemically altered. So PEP will give off its phosphate, releasing the energy which is required for the protein to pump the molecule that it wants into the cell. But as it moves it into the cell through the protein, it physically alters that molecule. So let's say we have glucose outside the cell, and we're going to move it into the cell by group translocation. That will use one PEP. It does use a transport protein. And then the glucose, when it's moving into the cell, will be changed from glucose to glucose 1,6-phosphate. So the phosphate from PEP will actually be transported to glucose. It's thought that this allows the cell to accumulate substances at higher levels than it would be able to do elsewhere. Let me try and go through an example here. So let's say we have 10 molecules of glucose outside the cell. The cell could move all of them by active transport, but let's say, let me make a column here. Let me see if I can make a table. Oh, shoot. I didn't get that to be what I wanted. Let me just try that again. So we have 10 glucose inside the cell. I mean, outside the cell. And currently inside the cell, we have none. Let me blow that up. Well, I have to blow it up this way because otherwise I'll get off where I have it. So 0 inside the cell. Now, if it were to transport all of them, well, let's say 9 of them inside the cell, and we have 1 outside the cell, we can move this 1 inside the cell, but that would be a very steep gradient because there's already 9 inside the cell. The cell can instead move 5 glucose. Let's go 5 outside the cell and move 5 inside the cell by active transport. And then these other 5 can be moved inside the cell by, well, let's go 4, by group translocation. Make another row here. Why isn't that working? There we go. So there's now only one glucose outside the cell. To move that one in, moving it into the cell, we are now going to move it against 4. Moving 1 against 4 is not as great as moving 1 against 9. And that means that the cell will have an easier time moving it into the cell. So that's one advantage of group translocation, and that is you can move molecules into the cell against its concentration gradient, but it will not be a real steep concentration gradient because half of them will be moving by active transport and the other half by group translocation. Any questions about any of that?
Based on your example, you
Uh-huh.
said it would be easier for it to go from the 1 to the 4.
Uh-huh.
Okay, thank you.
It's easier to move 1 against 4 than it would be to move 1 against 9. I guess we can put 5 there because eventually we'll end up with 5 inside the cell. And that way we'll move 5 by group translocation and 5 by active transport. Much easier than moving all 10 by active transport. Because that moving the last one, you'll be a real steep gradient. You'll be moving 1 against 9. Whereas here you're moving 1 against 4, which is just not as steep as 1 against 9. All right, let me close that. All right, so remember group translocation is similar to active transport, except it's only found in prokaryotes. It does use a different high energy phosphate compound. It does use a transport protein, that's similar. And then the molecule which is moved across the membrane is physically altered. Like glucose outside the cell, when it's moved into the cell, it'll change to glucose 1,6-phosphate. All right, let's talk a little bit about the cytoplasm. That is the substance inside the cell membrane. For prokaryotes, that's the entire inside of the prokaryotic cell. The cytoplasm is mostly water, but there are dissolved solutes in it. So about 80% of the cytoplasm is water. We do have other things that are dissolved in it, like DNA is actually most of the time dissolved in the cytoplasm. Only in eukaryote cells, when the chromosomes are going through mitosis or meiosis, and then the chromosome condenses, will the DNA not be in solution. What happens is the DNA condenses and then the chromosome will precipitate out of solution. We do find other things in the cytoplasm, like the nucleoid. That is just wherever the DNA is in the cytoplasm. There's the ribosomes. They are just too big to go into solution. And so you can actually see them. Let me blow this up. There's the artist rendition, and then the dots over here are actually ribosomes. They're too big to go into solution. And then anything else the cell is accumulating in the cell, if it accumulates too much, the molecule will precipitate out of solution and then form an inclusion, also called an inclusion body. So an inclusion is just something the cell is accumulating, and there's so much of it that it comes together and then precipitates out of solution. And we call that an inclusion. And that looks like one there. An actual inclusion in an electron microscopic image. All right, the nucleoid region is a part of the cytoplasm. However, it stains a little different because the DNA tends to stain differently than the rest of the cytoplasm. But wherever the DNA floats around in the cytoplasm, that is the nucleoid region. There is no boundary between the cytoplasm and the nucleoid region, because there's no membrane separating them in a prokaryotic cell. Any question about any of that? So wherever the DNA starts to float, that's the nucleoid region. And some region which is not where the DNA is, that is not the nucleoid region. You can have DNA in a prokaryote. Most of it is in one chromosome. So most prokaryotes simply have one chromosome. But there are some prokaryotes, and we're just talking about some cells, doesn't matter on the species, that they may have a small molecule of DNA, and it does have proteins in it, but it's very small compared to the chromosome, and these we call plasmids. And we'll talk about them more when we talk about genetics in Chapter 8. Any question about any of that? All right, I've already talked about ribosomes, so I'll move on. Ribosomes are the site of protein synthesis. Ribosomes are made up of both proteins, it's actually ribosomal proteins, and RNA, and we call that ribosomal RNA or rRNA. Prokaryote ribosomes are a little smaller than eukaryote ribosomes, and you should know that prokaryote ribosomes are 70S. Have I mentioned S before? The S stands for the sedimentation coefficient, where the prokaryote ribosomes sediment in the ultracentrifuge gradient at the 70S position. The eukaryote ribosomes are a little bigger, and they are 80S in size, and they sediment a little lower down in the ultracentrifuge gradient. You do not need to know that the ribosome has multiple units and that the large unit gets together with the small unit to make the complete ribosome. I'm not going to quiz you on that, so you don't need to memorize this part of the slide. All right, I've talked about inclusions. They are a reserve deposit within the cytoplasm. And when this reserve becomes so much that the molecules accumulate, they will just precipitate out of the cytoplasm. And when they do, we then call it an inclusion, because you can see the inclusions under the light microscope. The cell often will use an inclusion when resources are low, but it really depends on what the inclusion is. There are different types of inclusions, as shown here. You don't need to know these different types, so don't memorize this portion of the slide. But you should know that there simply are different types of inclusions. Actually, I'll go ahead and keep that here. Here's inclusions that are called magnetosomes. Here are inclusions that are called sulfur granules. And this cell is a cell that metabolizes sulfur. And so the cell will want to pump any sulfur it finds outside the cell inside the cell. And the sulfur will get high enough in concentration that it'll start precipitating from the cytoplasm, forming the sulfur granules, shown here. And then these are metachromatic granules, which you can barely see in yellow. This is the cell here. And there's 3 metachromatic granules in both of these cells. Any question about any of that? All right. The next cell structure I want to talk about that some, actually only bacteria have, are endospores. Most endospores are from 2 families, the family Bacillus and the family Clostridium. So all members of these 2 families make endospores. An endospore is not a reproductive structure. One parent cell makes one endospore when the environment is deteriorating. When the environment deteriorates enough, the parent cell will then die and the endospore will continue. So one cell makes one cell, and that's not reproduction. Any questions about any of that? All right, endospores are specialized resting cells produced mainly by Bacillus and Clostridium. There are a few other bacteria that make endospores, and I think there's one member where it's a family, but there's only one species in the family, and I forgot the name. And then there's another family where there's 1 or 2 species that make endospores. So most are from Bacillus and Clostridium. Endospores are highly resistant to desiccation, heat, various chemicals, radiation, and they can generally survive conditions that the vegetative cell cannot. The endospore has very little, if any, metabolism. When the endospore forms, we call that sporogenesis, where a cell is making an endospore, which actually this cell here is making an endospore. And that's also called endospore formation. When the endospore is free, there we go. Let me blow this up. So right there, we have a free endospore. When there's another free endospore. When the environment becomes favorable, the endospore can then germinate and give rise to a vegetative cell. And the vegetative cell will have normal metabolism and then undergo reproduction. Whereas the endospore itself does not have normal metabolism and does not reproduce. The endospore just rests, staying alive until the environment changes. And then it will germinate and return to in this case, a bacillus, which I guess right there would be an example. Any questions about any of that? All right, let's talk a little bit about endospore formation. The cell normally divides its DNA in the usual manner, and then one DNA molecule or one chromosome moves to one side of the cell, the other one moves to the other side of the cell. In this case, doesn't move all the way over, and that's because the cell will then begin to divide, but the cell does not divide evenly. The endospore portion will be smaller than the rest of the cell. The plasma membrane will then start to separate the DNA. The plasma membrane will then become 2 plasma membranes or 2 membranes. The peptidoglycan layer will then form between the 2 membranes. And then the spore coat will be put on. which I think this is the spore coat here. Oh, come on. This region here, but it could be just that region there. The spore coat is a hard surface and it retains most of the moisture inside the cell. And then when the environment deteriorates further, this parent cell, come on, which made the endospore, will die, and that will release the endospore. Any questions about any of that? A little video, which isn't that good, but does show endospore formation. Well, that's not running. We have a little commercial.
There are many reasons, and I just like to say one very important reason why I believe and think.
All right, here's the cell, and there it's making the endospore. Any questions? So remember, an endospore is not reproduction. It is a cell survival strategy. All right, here we have the structures of the eukaryotes. And I think we only have 20 minutes left, a little less. And so I'm not going to talk about the eukaryote cell structure because that should be just a review of your previous biology course. I do have the slides here. You can go through it. But I do want to talk about the endosymbiotic theory. The endosymbiotic theory is a theory to explain how mitochondria and chloroplasts came about in eukaryote cells. And so that's the evolution of chloroplasts and mitochondria. How did these organelles originate? First, these organelles are very different from all other organelles. One, they have two membranes, an inner and an outer membrane. They also have a DNA molecule within the membrane-bound organelle. So how did these organelles originate? The theory is that we had a proto-eukaryote which engulfed a bacteria. So the proto-eukaryote cell was much larger than the bacteria, and it just consumed a bacteria. I'm going to make it a Ron. And so that cell was eaten and taken in by the cell. And if it was eaten and taken in by the cell, then we'd have the bacteria here. And that would be its cell membrane and cell wall. I'm not going to draw the cell membrane separate from the cell wall, but it would be in a food vacuole. So it will be surrounded by a membrane. And this would be the vacuole or vesicle that brought the bacteria into the cell. And for some reason, this proto-eukaryote, it's before eukaryotes. It doesn't have a mitochondria, but this proto-eukaryote, for some reason, did not immediately consume that bacteria. And so let's put this here. This is the proto-eukaryote. I'm just going to abbreviate that P from now on. And then this is the bacteria, which I'll probably move over though. All right. The proto-eukaryote had glucose in its cytoplasm. Let's go here. And some of that glucose was moved by probably active transport into the bacteria. Could have done actually even by facilitated diffusion, depending on the concentration of the glucose here. The proto-eukaryote could make ATP. Oops, let me erase that. The proto-eukaryote could make ATP by fermentation. So that means one glucose will give 2 ATP. Oops, didn't do that right. However, the bacteria could engage in aerobic respiration, which I know we haven't talked about yet. And that means that for one glucose, it could make 38 ATP. So the bacteria took the glucose and then made ATP. And for some reason, the bacteria released some of that ATP and it moved into the cytoplasm of the proto-eukaryote. We don't know how much it was, but just some of the ATP moved into the proto-eukaryote. Any questions about that? And part of the reason why this may have happened, both the glucose moving to the bacteria and the bacteria making the ATP and then the ATP moving into the proto-eukaryote, was probably just because life was simple back then and molecules were moving around and the cells weren't really controlling them. As long as they had access to them, they used them. The proto-eukaryote then wondered what was going on. I lost one glucose, but I gained more ATP than I could make from one glucose. And so the proto-eukaryote kept the bacteria and then fed it glucose to get ATP. And that's because the bacteria could make more ATP than the proto-eukaryote could make from one glucose. Any questions about any of that? And so the bacteria and the proto-eukaryote, lived in symbiosis. The bacteria got a home, it was fed glucose, and then the bacteria benefited the proto-eukaryote because it made ATP for the proto-eukaryote. Nowadays, the theory is that the bacteria is not found anywhere else free living. It's only found in the proto-eukaryote cell, and we're going to change that from proto-eukaryote now to eukaryote. Because this bacteria inside the proto-eukaryote is now the mitochondria. And that's the theory about how eukaryotes got a mitochondria. A long time ago, they swallowed a bacteria and then started living in symbiosis with the bacteria. feeding that bacteria glucose, and then the bacteria made ATP for the cell, the eukaryote cell. Let's look at some evidence for this theory. Where am I? Oh, there it is. All right, the first evidence for the symbiotic theory is that we have some protists that actually will consume another smaller organism. Like here, it's a bacteria. This is a photosynthetic bacteria. And then this paramecium lives in symbiosis with the bacterium, giving the bacterium home, giving the bacteria the ingredients it needs, mostly water and CO2, but some minerals as well. And then the bacteria engages in photosynthesis, and some of that glucose that the bacteria makes will be given to the paramecium. We also have some amoebas which lack mitochondria, and those amoeba will engulf a bacteria, and then we'll use that bacteria, give the bacteria glucose, and then the bacteria will engage in aerobic respiration, making ATP, and share some of that ATP with the amoeba. So we actually have a few protist cells that do engage in engulfing smaller cells, bacteria, and then live in symbiosis with those bacteria cell. For the amoeba, it makes it ATP. For this paramecium, the bacteria gives it glucose. Furthermore, if you look at this cell, and this is actually two cells here, it's going through reproduction, will be in a food vacuole, so that it should have both its cell membrane and then a food membrane vacuole around it. And if we look at the mitochondria, let me go back to my picture here. The mitochondria has two membranes, an outer membrane and an inner membrane. And the bacteria that was originally engulfed would have the cell membrane and then the food vacuole membrane. So both would have two membranes. That's just weak evidence, but let me go through more of the evidence. When we look at the DNA of eukaryotes, the DNA is linear. There's multiple chromosomes. When we look at the DNA of prokaryotes, it's generally one circular chromosome. When we look at the DNA of the mitochondria and the chloroplast, there's one circular DNA molecule. When we look at histones, eukaryote DNA has histones in the DNA. Prokaryote, I should say bacteria DNA lack histones in the DNA. And we look at the DNA of the mitochondria and the chloroplast, they lack histones. When we look at the first codon, what AUG codes for in eukaryotes, AUG codes for the amino acid methionine. So all proteins, when they first start, will have the amino acid methionine. In prokaryote cells or bacteria, it's a similar amino acid that AUG codes for, but it's a different amino acid. It's formylmethionine. And we look at the first amino acid in the mitochondria and the chloroplast, it codes for a formylmethionine. when we look at the ribosomes of eukaryotes, they're 80S in size. The bacteria have 70S ribosomes, and we look at the ribosomes found in the mitochondria and the chloroplast, they're 70S. Eukaryote cells will divide by mitosis. Prokaryote cells will mostly divide by binary fission. When we look at the mitochondria and the chloroplast, they divide by binary fission. And then the strongest evidence for the fact that the mitochondria and the chloroplast were at one time prokaryote cells is when we sequence the DNA of the eukaryotes, the DNA will fall in the domain eukaryotes. When we sequence the domain or the DNA of the prokaryote cells, or at least the bacteria cells, it'll fall within the domain bacteria. When we sequence the DNA of the mitochondria, the sequence of the DNA of the mitochondria falls within the domain bacteria. And the same with the sequence of the domain of chloroplast. The DNA falls within the domain bacteria. So the theory is that animal cells got one bacteria, which then eventually evolved into the mitochondria. And that cell, we don't call the proto-eukaryote anymore. We call it the eukaryote with the mitochondria inside of it. And then the plant cells got both the mitochondria, and then later on they got the chloroplast, and then they have two bacteria that they lived in symbiosis with to evolve into the mitochondria and the chloroplast. Any questions about the endosymbiotic theory? All right, that's the endosymbiotic theory to explain how eukaryotes got mitochondria and chloroplasts. Are there any questions at all on this lesson? If not, I'm going to end it here, and I'll see you at 6:30 for the lab. All right, bye.